Dr. Corey Smith from Queensland International Medical Research, QRMR, Brisbane, Australia. Dr. Smith currently is very active in doing research on herpes virus vaccine development and immunotherapy. And today he would like to share with us about the development of vaccine against viral-related cancer. 35 minutes. Okay. Please switch on the... Yeah.
That better?
Yeah.
Sorry. I'd really like to thank the organisers for the opportunity to come and present some of the work we've been doing in Brisbane. And so our real interest is in developing vaccines to treat herpes virus. And yes, first of all, I just would like to acknowledge a couple of people, Professor Rajiv Khanna and Professor Dennis Moss, who some people in the audience will actually know quite well. And these are the people who've really been driving our research, developing vaccines for herpes viruses. Is that scrolling by itself? Or is that me pressing the button? Okay, so sorry, I'll get through into this, sorry. So what we actually know is that approximately 12% of all human cancers are actually associated with viral infection. And 80% of these cancers are actually in the developing world. Now, what we actually know is that rather than being a symptom of primary infection, where you get infection with the virus and then you then go on to develop a cancer, what's really known is that these cancers take a long time to form. And this is during when these viruses actually go into a latent state in your body and then induce transformation events that may then lead to malignancies. Oh, sorry. So there's actually a number of viruses worldwide which are associated with cancer, and these are the 6 clearest examples. So this is just a slide showing the annual cancer incidence and the number of cancers associated with each of these viruses. So the primary cause of viral-associated cancer is actually human papillomavirus, which causes cervical cancer, but it also is now being associated with some non-melanoma type skin cancers, which are quite common in countries like Australia. Obviously, probably quite well known to the audience, the cancers that are associated with hepatitis viruses, and these are liver cancers. And this is also a similar sort of incidence between the two for the liver cancers that are associated with hepatitis B and hepatitis C. Now, our research interest is actually in Epstein-Barr virus. So Epstein-Barr virus is actually associated with approximately 200,000 cancers worldwide annually. And this is quite a range of cancers that can either be of epithelial origin, such as nasopharyngeal carcinoma, which has an increased incidence in Indonesia and other parts of Southeast Asia, but also other carcinomas, gastric carcinoma worldwide, and also lymphomas, which are probably which we may have a high prevalence of in Western countries, such as Hodgkin's lymphoma, but also some non-Hodgkin's lymphoma and Burkitt lymphoma, which is actually in Africa. And there are obviously a few other viruses associated with cancers. So what we really, I guess, need to understand when we're trying to develop vaccines against cancers is understanding really how the immune system interacts with these viruses and what we can actually target. So this is quite a very simple representation of how we'd see cancer formation occurring after these initial viral infections. So what we typically see, obviously, you go through a stage where you're exposed to the virus, you get lots of virus made, and then it's controlled by the immune system. And this is then usually controlled by neutralising antibodies, which are placed at mucosal surfaces, which will block reinfection, but also by systemic immunity by effector and memory T cells. You'll then go on to a stage of persistent latent infection, where the virus is permanently in your body. It may be replicating, it may be inducing just division of cells. And again, this stage, it's really effector and memory T cells that control this. Then what we think is you'll probably go through a stage of a pre-malignant infection and then move on to a malignant infection where you have the virus and that's associated with the malignancy. So what we really think is happening here is that there's a breakdown in immune control. So that there's a loss of the quality and function of these T cells. So this, we think, therefore gives us sort of 3 avenues where we can vaccinate against these cancers. The most obvious choice, and which has been successfully used for hepatitis B and human papillomavirus, is to develop a prophylactic vaccine to prevent infection. So this is where you target the production of neutralising antibodies. This prevents you from being infected, so you're expected then to prevent the downstream effects associated with the malignancies. But this we think is much harder for something like EBV. So the other options are to produce these vaccines that will target the induction of T cell responses, either in a prophylactic setting to prevent the formation of these malignant cells by controlling this persistent latent infection, or in a therapeutic setting by actually treating people who have a malignancy. So first of all, I'll just go briefly through how these prophylactic vaccines work. So what it's known, as I mentioned, these primarily target the induction of neutralising antibodies. And what neutralising antibodies target are the proteins on the surface of a virus. These are either glycoproteins or other proteins that are required for entry into the cell. So the best two examples of these and the licensed vaccines that treat viral-associated cancers at this stage of infection are those for human papillomavirus, and these are what's known as virus-like particles that encode these L1 and L2 proteins, and also the hepatitis B virus vaccine, which is again a virus-like particle that actually targets the hepatitis B surface antigens. So I just wanted to briefly go through some of the data which shows the effectiveness of this type of vaccination strategy in preventing infection and obviously potential side effects associated with this. So this is just looking at results from a recently published review on the effectiveness of the hepatitis B surface antigen vaccine. So this vaccination program has been introduced in well over 90% of countries, and for a lot of countries this is over 20 years old. So this is the incidence of hepatitis B surface antigen carriage, but before these vaccination programs were introduced in a number of parts of the world, particularly obviously in Indonesia and other parts of Southeast Asia, but also in some parts of Europe and Africa. And you can see the dramatic reduction in the rate of hepatitis B surface antigen carriage as a result of this vaccination strategy, clearly showing the effectiveness of these vaccination approaches. So the vaccines for human papillomavirus are a lot more recent and programs have only been introduced in the last 5 to 6 years in most countries. So these prophylactic programs are now implemented in over 40 countries worldwide. And Australia actually introduced the first program, which was in 2007, primarily because one of these vaccines was developed by groups in Queensland. Now, this is just some recent estimates of the efficacy of this vaccine, and this was from a publication from some researchers at QIMR who are primarily epidemiologists, but they could see a significant reduction in these high-grade cervical abnormalities that are associated with HPV infection. So clearly, in addition to reducing the risk of infection, you can reduce, obviously, the malignancies associated with these infections. Oops, sorry. So as I mentioned at the start, our primary interest is in EBV vaccines. Now, I just want to briefly go through EBV quickly. So EBV is actually a member of the gamma herpes virus family, and there's another member of this family which is also associated with cancers. This is the Kaposi sarcoma herpes virus is actually very common in Africa. Now, the worldwide prevalence of EBV is in excess of 90%. So most of the people in the room will be infected with EBV. And in a place like Indonesia, early childhood infection actually reaches up to close to 100% of the population. And what we know in Indonesia has an increased incidence of nasopharyngeal carcinoma, and there is some anecdotal evidence to suggest that some of this at least might be associated with early childhood infection. And in countries like Australia, the dynamics of infection is actually different, where we do see quite significant early childhood infection, but obviously at lower rates. And we then have a second peak of infection in adolescents and also in adults. Now, this second peak of infection in adolescence is actually associated with a syndrome in Australia and other developed countries known as infectious mononucleosis, which is an acute disease that usually fairly quickly resolving in most individuals, but this also increases the risk of Hodgkin's lymphoma. So the other thing I want to point out about EBV, it's a very large virus. So this contains in excess of 85 genes. It has a very complex life cycle. And if you compare this to viruses like HPV and HBV, which are very small viruses, which makes it potentially targeting it for this sort of prophylactic vaccine a lot more difficult. However, there have actually been attempts to develop a prophylactic vaccine for EBV, and this is primarily by targeting the surface protein GP350. So this is one of the glycoproteins on the surface of EBV, and it promotes entry into B cells. So B cells are the primary source of latent infection with EBV. And it's known that if you generate serum specific for GP350, it will block the entry of EBV into B cells in vitro. So there have been up to phase 2 trials looking at GP350 as a vaccine to prevent EBV infection, and this was primarily in seronegative healthy adults. And what was interesting is while this vaccine showed efficacy in preventing primary infectious mononucleosis, these are the diseases associated with acute infection, which might then go on to lead to Hodgkin's lymphoma, there's really no efficacy in preventing asymptomatic infection. So there was no protection at all from people getting infected. And this sort of vaccine, you're really difficult to tell whether it would be efficacious in reducing the risk of nasopharyngeal carcinoma. And the reason for this is primarily that we don't really understand how EBV infects. So there's kind of two schools of thought how EBV passes. It's obviously acquired orally. And these schools of thought are either directly infects B cells in the oral mucosal epithelium, or during this primary lytic stage of infection, it infects via a different route directly into epithelium. So this is quite a potential complex route of infection, which probably makes it very difficult to block this primary infection. But what we then do know is following this infection, EBV drives this transformation of B cells, which are these B cells survive for life with EBV in them, and then this can be then spread and shed into other, to infect other individuals. So what it's clear is there's clearly there potentially a need to develop, rather than a prophylactic vaccine, develop a therapeutic vaccine that would allow the targeting of EBV. So as I mentioned, there really isn't any current licensed prophylactic vaccine for EBV, and we really don't think there's going to be one in the near future. But also point out, obviously there's a lot of chronic carriers of hepatitis B virus worldwide. This is in excess of 240 million. So clearly the potential, there's still some need for finding solutions that may be vaccine-based to treat these patients, as well as the fact that the HPV vaccines are limited to 2 serotypes, which are the major cause of cervical cancer worldwide, but there's obviously another significant potential for cervical cancers from other serotypes. And I also want to point out is the current vaccines to HPV and HPV don't prevent cervical cancer or liver cancer associated with HPV, because they're unable to stop the life cycle once it escapes into the virus. So what I'm going to talk about today is our strategies to develop vaccines for EBV-associated malignancy. So as I mentioned, this is the basic route of EBV infection, where it induces latency in these B cells, and these B cells survive for life. And what we also know is that, probably flip too far, is that what controls this stage of infection is EBV-specific T cells. So these T cells provide permanent surveillance in most people, and you have an asymptomatic infection for life. You don't go on to develop other diseases. But what we do know is you can get malignancies associated with these stages of infection. So this includes the malignancies you can see in B cells, which are the Burkitt's lymphoma and Hodgkin's lymphoma and the post-transplant lymphomas, but also malignancies actually which are associated with this epithelial infection. So these are the nasopharyngeal carcinoma, which is common in Indonesia and other parts of Southeast Asia, but also gastric carcinoma. And what we think is going on is some breakdown in the immune surveillance of these malignant cells that allows malignancy to escape. So normally you would expect, because you have a viral antigen expressed in these cells, that you would get control through the induction of immune response, but this doesn't occur. So it's also known about these malignancies is that they vary in the pattern of EBV gene expression. So the latent life cycle of EBV is characterised by the expression of approximately 8 genes out of those 85 initial genes, and these vary as the virus spreads and establishes latency in their level of expression. But this also vary in the malignancies that arise as a consequence of these infections. So what we know in the malignancies that arise in immunocompromised patients, these are patients where their immune system really isn't working, they don't have T cells, you get these malignancies which express the whole array of EBV latent genes. These are these EBV1 and 6 and these LMP1 and 2 genes, and these are quite easily targeted by immunotherapeutic approaches because these are very immunogenic. What we know in these other malignancies is that you get a restricted pattern of EBV antigen expression. And what was known initially in Burkitt's lymphoma, which is where EBV was discovered, that you only see expression of one of these genes associated with latency. And what we also know has become known in malignancies such as nasopharyngeal carcinoma and Hodgkin's lymphoma. You get this classically type 2 profile where you're expressed these 3 latent genes, and then in these other malignancies, which have been more recently discovered, you get an even more restricted pattern of expression. So what it's clear, we think, is that these genes are important for malignancy in these immunocompetent settings. Now, what's interesting, this was from a recent publication looking at the interaction between viruses and how they may promote some of what are considered the hallmarks of cancer. Now, I won't go through this in detail, but these are considered the hallmarks of cancer, what cancer cells have to do to survive. And what we know is that some of these viral genes actually promote these survival mechanisms. So the key functions of these proteins, so we know from LMP1 it's required for B cell transformation, for the B cell to survive and proliferate, and it may be similar in epithelial cells. So we know that it's providing this sustained proliferative signalling. We know from LMP2 it's an anti-apoptotic molecule that allows B cells to survive when they're infected with EBV, and they won't apoptose. So LMP also has some role in promoting these hallmarks of cancer. And we know from EBNA1 that its primarily role is to maintain the EBV episome. So as the B cell divides, the EBV divides without needing to replicate. And this is also promotes some of these hallmarks of cancer. One thing I want to point out is these antigens are very good at avoiding immune destruction, which is important, and particularly with recent evidences in a lot of cancer showing that if you target the immune system to recognise these malignancies, you can get actually wonderful survival outcomes. But what we know is that these antigens are very good at avoiding immune recognition. So, and I just wanted to take you through a little bit of work we've actually done to look at this and as to why we could theoretically target these antigens to actually boost immunity to these antigens. So what we know is if we take blood just from healthy laboratory donors, so this is possibly from anyone in the room or anyone at our research institute, is that your immune response, so this is just looking at the ability to make cytokine in response to these antigens, your immune response to EBV is primarily directed against the antigens of the lytic cycle or these EBNA 3 to 6 antigens, which is why obviously the post-transplant lymphomas which express EBNA 3 to 6 only arise in people whose immune system is suppressed. But what we know is you induce very poor immune responses against LMP1 and 2 and EBNA1. And there are very specific reasons for this that I won't go into today. But it's clear that your immune system is poor at priming an immune response. So what we've also gone on to show is that these T cells are not very functional, that they really are poor at recognising antigen and potentially controlling antigen-infected cells. And this is an assay where we can look at the ability of these T cells to make multiple cytokines. So initially this idea came out of work looking at HIV patients, where they could show in those patients that were persistently infected but didn't really develop into AIDS, so those long-term survivors, they had these what were called polyfunctional T cells that are really good at controlling viral infection. And this is just 3 cytokines we use in this assay. You can do this with other molecules. But what we see this pattern in these lytic T cells where you get a high proportion. So these are individual donors assessed against healthy donors where you can see a strong response against making 3 or 2 cytokines, whereas what we see in our LMP1 and 2, but also in our EBNA1 T cells, is that these T cells are really poorly functional, that they don't make very many cytokines. Particularly, they don't make a lot of interferon gamma, which is a classical measure of CD8 T cell function. So just a few other things to go on with. What do we think are the consequences of this? Well, we think the consequences of this are that these T cells are more susceptible to immune regulation, which may be mediated by the tumour. So this was in a study we actually did in Hodgkin's lymphoma patients with a cohort throughout Australia. And this is just looking at their immune response when they have the acute malignancy as opposed to when they're in remission. So what we can see is a specific, and this is actually using an ELISpot assay, again looking for interferon gamma secretion. We can see a specific loss of function here in this pre-remission phase, and then you can see a recovery in this response. And you don't see this change at EBNA 3 to 6 T cells or lytic T cells. Again, showing that this is some changes in these T cells that mean they probably struggle to recognise these malignant cells. And what we've then gone on to show is to show a specific mediator of this, at least relevant to Hodgkin's. So what we can see is that this is a molecule called galectin-1, which is produced by a lot of cancers, which can suppress immune function. And in these settings, what we can see is that we can suppress the proliferation of these LMP1 and 2 specific T cells. So this is an assay where you just label with a fluorescent dye CFSE, and then you give them antigen, and when they're stimulated, they divide. So you can see what we can see is suppression of these LMP1 and 2 specific T cell function, but we can't see this suppression in our EBNA 3 to 6 or in our lytic specific T cells. So what we really think is going on is that the weak immunogenicity of these LMP1 and 2 and EBNA1 of EBNA1 contributes to the poor immune surveillance of these EBV-infected malignant cells. So our hypothesis really is that if we improve the immunity to these antigens, this will lead to an enhanced immune surveillance and recognition of these malignant cells. And obviously, we think the ultimate best way to do this is by developing an immunotherapeutic vaccine which would specifically target this LMP and EBNA1 specific immunity. So this is the technology we've actually developed to do this. And this is what we call E1 LMP poly, and this is known as multi-epitope technology. So what this basically involves is identifying regions within these proteins that are presented to cytotoxic T cells, which in this instance are from LMP2 and LMP1, and this is across HLA types which would cover globally. So provide us with the opportunity to use this sort of strategy across a diverse array of ethnic groups. And in this instance, so these are just strung end-to-end in what we call a polyepitope, and in this construct, this is fused to EBNA1, truncated form of the EBNA1 gene, which provides us potentially with CD4 responses as well as CD8 T cell responses, and we encode this in an adenoviral vector. So the reason we actually do this is because we think it increases the immunogenicity of these epitopes, and it also takes away the oncogenic potential of those proteins, which have the ability to induce transformation. So I'm just going to go through a little bit of some of our preclinical data and then go into some of the clinical studies we've been doing using this vector. So this is just looking in human HLA transgenic animals, where we can look either during effector priming or after long-term priming. And this is just looking at the immune response we can generate to the LMP2 and LMP1 antigens. You can see significant expansion. If you compare that to the responses we see in humans, it's obviously a lot more significant, and this is relative to our controls. So this is just the overall analysis in all of our animals in this study, and again, obviously you can see strong responses. But what we can also see is we can introduce now, following this vaccination, is this polyfunctional profile in these T cells. So you can see primarily what we're now getting is a lot of cells making interferon gamma. And in this instance, we've included actually another measure of T cell function, which is degranulation. And you can see now we have majority of our cells are either making interferon and CD107 and TNF, or are making this combination of interferon and CD107, or they're making CD107 alone. So this is suggesting with the vaccination we can induce qualitatively better T cells. So ultimately, we'd like to take this vaccine into the clinic, but before we do that, we've actually decided on assessing another approach, which is a slightly more complicated approach, but it's been used successfully in slightly different circumstances to treat post-transplant lymphomas. And this is known as adoptive T cell therapy. And this is how our adoptive T cell therapy works. And this is primarily so far in nasopharyngeal carcinoma patients. So basically what we do is we get a bag of blood from these patients. We can then expose the T cell or the white blood cells in their blood to our viral vector, and this is then mixed with white blood cells. And what happens from these other white blood cells, we get an expansion of these cytotoxic T cells, and then we reinfuse them into our patients. So to carry out our first clinical study with this, we're actually, obviously in Australia, we have a very low incidence of NPC. We do have some NPC, particularly from immigrant communities from China and Southeast Asia. But the highest incidence of NPC actually occurs in Hong Kong and in southern China. So we've looked to Hong Kong to establish this principle that we can use adoptive immunotherapy potentially to treat NPC patients. So this is just the outline of our first study which we've completed. So this involved recruitment over a 3-year period. And altogether we recruited 25 NPC patients. So whilst they had varying disease at their initial stage, at the time of consent, most of these patients had late-stage disease. They were refractory to normal chemotherapy. They were obviously very close to passing away. And we had one patient who had a local recurrence. This is something we're quite interested in treating patients earlier in disease progression. We think that's a better option for T-cell activation. So this is just to show that what we can do in mice with expansion of antigen-specific T cells and generating polyfunctional T cells, we can also do in these NPC patients. So again, this is in vitro by growing these T cells in a tissue culture flask. But this is from one of our patients. And again, you can see ex vivo, we see this poor function with very difficulty detecting these T cells. And what you can see post-expansion, we can see the generation of these polyfunctional T cells, showing that we can do a similar thing for human-specific T cells. And it'll go back. No, it won't go back. Okay, we'll skip that slide. That should just show that we can generate therapy for most of our NPC patients. Sorry about that, I don't know what happened. Okay, but what we can see generally in these safety trials, phase 1 trials, it's a safety trial. We need to look at whether this is a safe therapy. And always in the case with these autologous therapies, they're usually very safe. We see very little side effects, with a few flu-like symptoms and malaise, but generally these are very safe. So this is just, we actually managed to treat 14 patients. We manufactured therapy from 16, and actually 2 were too sick prior to the ability to treat. And this is looking at disease progression after treatment. While these results may not look that impressive, so what this means is that the time point at which each of these patients progressed after we gave them their adoptive T-cell therapy. So what you can see is, what we saw in the majority of these patients is some stabilisation of disease, where we could potentially block the tumour from growing, but eventually all of these patients progressed. And we think this is the difficulty in treating patients at such late stage, with such large tumours, with lots of metastases. And what we've also looked at in this cohort is looked at overall survival. And there's actually a few caveats in this data. This is looking at our treated versus our untreated patients. But our untreated patients, this is not a properly controlled study. It's just to show you that we look like we improved overall survival in the patients we treated. But as I said, the actual caveat in this is that these untreated patients may have been sicker in the first place, and that's why they died more quickly. But what's also evident is this overall survival was an improvement on the institutional controls. But obviously we'd like to take this further into later stage trials. So what I want to show you is where we're actually moving now. And this came about from this patient. So this is a patient who had a really large tumour before immunotherapy, and this tumour was actually refractory to any chemotherapeutic options that could be offered. And this is 4 weeks after we give them the T-cell therapy. So what you can see is the tumour's static, but we really didn't see any signs of regression, which was interesting. And then they actually progressed and were withdrawn, and then they got 1 cycle of chemotherapy. So you can see this dramatic reduction in the tumour load, suggesting that there's something that's happening which is a combination of the immune therapy and chemotherapy. We don't quite understand exactly what's going on, but it may be potentially some sort of what they call debulking that allows the chemotherapy to get in, but we don't know. Unfortunately, this patient did go on to progress again and relapse. But what we've also been doing is in a smaller cohort, this is actually primarily in Brisbane with a small cohort, is looking at if we introduce this immunotherapy into patients we think of high risk of relapse. So these are patients that have had surgery or chemotherapy, and then we're looking at then introducing immune therapy during the stages of remission. Oh God, that's killing me. Okay, sorry. And what you can see is, what we also see is we can get much better T-cell expansion in these patients. All of these patients show overall progression-free survival. And interesting, just the little data from one of these patients is that we could see potentially some control in EBV load. So EBV load is used as a measure of viral load in this setting. And what's interesting, although this is a long time after immunotherapy, we could see that while they see a peak in EBV load, this was actually reduced. Potentially as a result of immunotherapy, we can't say for sure. This may have been some other mechanism of controlling this potential increase in tumour. So this is actually where we're going to from now. We want to really move our immune therapies forward, whereby previously we were treating late-stage patients, but what we're now trying to do is to treat these patients at earlier stage. And this will be in patients who have relapse metastatic disease, but primary relapse in metastatic disease. So what we will get is blood from these patients before they start the chemotherapy. We then grow T-cells. Whilst these T-cells have been grown, the patient will receive their normal chemotherapy, and then we will deliver the immunotherapy. Now this won't be a controlled trial, but it's actually moving slightly forward in our phase trials, but it's still a safety trial because this is in a different cohort. But what we'd really ultimately like to do is to use this as a vaccine. So this is the same vector that we think would be effective using as a vaccine. And the adenovirus vaccines have actually been shown to be fantastic in humans at inducing T-cell responses. So really what we want to do is to chop out all that middle work, and after the patient gets chemo, we just inject the vaccine and prime T-cell responses. But this is a little bit more complex, which is surprising, and we obviously need to have collaborators, actually industrial collaborators who are interested in developing this sort of approach. But this sort of approaches have been tried by other groups, I must say. And this is just some data I'd like to present at the end from one of these groups, which has trialled a vaccine approach. So this is another group in Hong Kong in collaboration with groups in in the UK. And this is where they use these are modified vaccinia, which is the vector used for smallpox vaccination. And then they introduce into that the LMP and the EBNA1 antigen, the LMP2 and the EBNA1 antigens. So what they can see, this is just one figure out of their publication showing one of their patients. And what you can see is they can see a peak in their T-cell response, and this is a CD8 T-cell response, but also a CD4 T-cell response, but this does decline, and this is not actually a strong response. We think there are some vectors that are much more effective in humans at inducing CD8s. But what was interesting, which kind of parallels what we saw in our adoptive therapy study, is that you can see this control of EBV tumour loads in the plasma. Well, this is obviously a little bit after they give their vaccination, but it suggests maybe that priming these T-cells can recognise the cells that are EBV infected, whether they're malignant or not. But eventually this patient, as happens in a lot of patients, they went on to relapse. But it's interesting. It suggests that vaccination might be a viable approach, and we'd like to explore that in the future. So just in conclusions, what is clear is that the prophylactic vaccines are the best way to treat these viral-associated cancers. By preventing infections, you're obviously going to prevent the cancer. But what we think from EBV is this is really going to be a very difficult approach. We don't think you can induce sterilising immunity. And this is going to face significant hurdles, particularly in populations where 100% of people are infected in the first 5 to 6 years of life. And then for most people, it's an asymptomatic infection for life. You don't see any symptoms. But what I've also hope I've shown you is that it's really the oncogenic viral proteins, once this latency is established, that you need to target in any vaccine approach. And we think the best way to do this is with inducing cytotoxic T cells. Okay, so finally I'd just like to thank the people involved in this study. Obviously I've also already mentioned Rajiv Khanna and Dennis Moss. So Dennis has actually probably been working in EBV for over 40 years, and it's actually the 50th year of EBV's discovery this year in Burkitt's lymphoma. So he has a long history of working not only in NPC research but also in Burkitt lymphoma, which is actually endemic in parts of Papua New Guinea, and I'm sure potentially in places in Papua potentially as well. And also, obviously, the ability to manufacture these T cells requires a lot of hands-on, and we've had a large number of members of our group who were involved in clinical coordinating these trials, but also in manufacture, and also, obviously, importantly, our clinical collaborators, particularly our collaborators in Hong Kong, which help us conduct this kind of proof of principle trial, this safety trial, but also some collaborators in Brisbane who are involved in recruiting some local patients and looking at immunotherapy in what we consider a high-risk cohort. Thank you.
Thank you very much. Very on time. Yeah.